Welcome to Module 1 of our course on Industrial Biochemistry. In this session, we will set the foundation for your understanding of how living systems, particularly microorganisms, are harnessed for industrial processes that produce food, fuels, chemicals, and pharmaceuticals. Industrial biochemistry is, in many ways, the marriage between biology and industry. It takes the deep biochemical understanding of life at the molecular level and applies it to large-scale production systems. It is not enough to know that microbes can ferment sugars. We must also understand how to control and optimize these processes so that they are economically viable, safe, and efficient. In this module, we will start with a review of microbial physiology and genetics, then revisit general metabolic pathways with a focus on their industrial applications. We will explore continuous culture methods, the principles behind them, and how the chemostat has become an indispensable tool in industrial fermentations. We will also cover the major types of fermentation, alcoholic, amino acid, antibiotics, and other secondary metabolite production, linking the science of metabolism to the engineering of production systems. By the end of this module, you should not only know what happens inside an industrial fermenter, but also understand the biological rationale behind each step and how these processes can be manipulated for better yields. 1. A short review of microbial physiology. Let us begin with microbial physiology, the study of the life processes of microorganisms. In industrial biochemistry, our key players are often bacteria, yeasts, and filamentous fungi, although certain processes also employ algae or protozoa. Cell structure. Bacteria are prokaryotes, meaning they lack a true nucleus and membrane-bound organelles. Their genetic material is a single, circular chromosome located in the nucleoid region. Eukaryotic microbes, such as yeast, have a nucleus and membrane-bound organelles, allowing for more complex compartmentalization of metabolic processes. Growth and nutritional requirements. Microbes require sources of carbon, nitrogen, minerals, and sometimes growth factors. Carbon sources may be sugars, alcohols, organic acids, or even hydrocarbons. Nitrogen may be supplied in inorganic forms such as ammonium salts or nitrates, or in organic forms like amino acids and urea. Industrial biochemists must understand these needs to design optimal media for large-scale fermentation. Microbial growth phases. In batch cultures, microbial growth typically follows a pattern. Lag phase, cells adapt to the new environment, no immediate cell division. Log, exponential, or phase, rapid cell division, high metabolic activity, the prime time for production of certain metabolites. Stationary phase, nutrients become limiting, waste products accumulate, and growth rate slows. This phase often triggers the production of secondary metabolites such as antibiotics. Death phase, cell death exceeds cell division due to unfavorable conditions. These phases are crucial to understand because industrial processes must often be timed to harvest products when yields are optimal. Enzyme systems. Microbial physiology also deals with enzymes, the catalyst of biochemical reactions. Enzymes can be constitutive, produced continuously, or inducible, produced in response to specific substrates. In industry, controlling enzyme production is a way to control the rate and specificity of metabolic pathways. 2. A review of microbial genetics. Genetics determines the capabilities of microorganisms, what they can metabolize, what products they can produce, and how they respond to environmental changes. DNA structure and replication. Microbial DNA contains genes that encode proteins and RNAs essential for survival and production. During cell division, DNA replication ensures that genetic information is faithfully transmitted to daughter cells. In biotechnology, manipulating microbial DNA allows us to enhance production of desired compounds or introduce entirely new metabolic abilities. Gene expression. In prokaryotes, gene expression is often regulated in operons, clusters of genes under the control of a single promoter. The lac operon in E. coli is a classic example, switching on lactose-metabolizing enzymes only when lactose is available and glucose is absent. In industrial applications, gene regulation can be manipulated to overproduce specific enzymes or metabolites. Genetic variation. Variation can occur naturally through mutation, recombination, or horizontal gene transfer, via transformation, transduction, or conjugation. Industrial microbiologists may induce mutations artificially to generate strains with higher yields, faster growth rates, or resistance to inhibitory byproducts. Recombinant DNA technology. The integration of genetic engineering with industrial biochemistry has revolutionized the field. For example, genetically engineered yeast strains can produce higher ethanol concentrations without being inhibited by alcohol toxicity, or bacteria can be engineered to secrete enzymes directly into the culture medium for easier purification. 3. General metabolic pathways and industrial applications. Metabolism is the sum total of all chemical reactions within a living cell. It is typically divided into catabolism, the breakdown of molecules to release energy, and anabolism, the synthesis of complex molecules from simpler ones, requiring energy. Central carbon metabolism. Glycolysis, the breakdown of glucose into pyruvate, producing ATP and NADH. TCA cycle, Krebs cycle oxidizes acid-CoA to CO2, generating NADH and FADH2. Oxidative phosphorylation uses NADH and FADH2 in the electron transport chain to produce ATP. In industry, these pathways are not only sources of energy but also of precursor metabolites used to build amino acids, nucleotides, and secondary metabolites. Fermentative pathways. When oxygen is limited, cells switch to fermentation. Depending on the organism, fermentation can produce ethanol, lactic acid, butanol, or other products. For example, Saccharomyces cerevisiae ferments glucose to ethanol and CO2, while Lactobacillus species ferment sugars to lactic acid. Industrial relevance. Understanding these pathways allows industrial biochemists to modify conditions, such as oxygen supply, pH, and nutrient composition, to favor the production of a desired metabolite while minimizing unwanted byproducts. 4. Continuous culture methods. In contrast to batch cultures, where all nutrients are supplied at the start and products are harvested at the end, continuous cultures maintain microorganisms in a steady state by continuously adding fresh medium and removing an equal amount of culture. Advantages. Constant supply of product. Microbes can be maintained at a specific growth rate for long periods. Easier control over environmental conditions. Types of continuous culture systems. Chemostat, fresh medium is added at a constant rate, and culture is removed at the same rate, keeping the culture volume constant. Growth rate is controlled by the dilution rate. Turbidostat uses optical density sensors to maintain a constant cell density by adjusting the flow rate of fresh medium. Continuous cultures are particularly valuable in the production of single-cell proteins, organic acids, and certain pharmaceuticals, where stable conditions can maximize yield and quality. 5. The chemostat and its application in industrial fermentations. The chemostat is perhaps the most widely studied continuous culture system in industrial microbiology. Its defining feature is that the growth rate of the microorganisms is set by the rate at which fresh medium is supplied. Key principles. The growth-limiting nutrient in the medium determines the maximum biomass that can be maintained. At steady state, the specific growth rate of the culture equals the dilution rate. Industrial applications. Selection for specific traits. By setting nutrient limitations, we can favor organisms that are most efficient at utilizing that nutrient. Studying microbial physiology under defined, steady-state conditions. Continuous production of metabolites that are best formed during exponential growth. For example, the production of certain amino acids may be optimized in a chemostat by maintaining cells in a growth phase where precursor metabolite pools are highest. 6. Overview of fermentation processes. Fermentation, in industrial terms, refers to any large-scale microbial process for producing a desired product, whether or not it is strictly anaerobic. Alcoholic fermentation. Conducted mainly by yeast such as Saccharomyces cerevisiae. Converts sugars into ethanol and CO2. Used in brewing, winemaking, bioethanol production. Amino acid fermentation. Bacteria such as Corynebacterium glutamicum and Escherichia coli are used to produce amino acids like lysine, glutamic acid, and threonine. These are essential in animal feed, food seasoning, e.g., monosodium glutamate, and pharmaceuticals. Antibiotic fermentation. Many filamentous fungi, Penicillium chrysogenum, and actinomycetes, Streptomyces species, produce antibiotics as secondary metabolites during stationary phase. Industrial processes involve careful control of aeration, pH, and nutrient limitation to maximize yield. Other secondary metabolites. Includes pigments, alkaloids, and immunosuppressants. Often produced under nutrient-limited, stress conditions, requiring precise timing in batch or fed batch cultures. At this point, we've covered the physiological and genetic basis, the metabolic pathways, and the engineering approaches including continuous culture and chemostat used that underpin modern industrial biochemistry. The second module will build on this by exploring applications, strain improvement, and advanced industrial strategies.